Okay, so a pleasant good day to all. And today we'll be looking at chapter 3, cells and tissues, and see how they relate to the general scope of things within the biological realm. Now, one of the important things as it relates to cells and tissues, it shows the very important relationship as it goes or relates to the form and function. What do I mean by that? Well, when you look at a neuron is a specialized nerve cell which is designed to collect impulses from other nerve cells. Hence, when you do look at the structure of a neuron, you see the top part of the neuron has numerous dendrites or things that look like antennae to interact with other dendrites from other cells to receive these inputs. So very importantly, structure-wise, neuronal cell is very well adapted in terms of the regions around the cell body to accept impulses or input from other cells. Similarly, when we look at the structure of an intestinal epithelial cell, we would notice numerous villi present on the surface of the cell itself. And these villi, or these extensions of the cell, they increase the surface area, thereby increasing or enhancing the cell's ability, in this case, the intestinal cell, to absorb nutrients. So this is very important. Intestinal cell, neuronal cell, two adaptations, which shows the importance of structural changes as it relates to the functionality of the cell itself. Okay, so another part of the cell membrane, this is an enclosure around that encompasses the cell. It could be likened to a fence around your home. So what the fence does, it keeps things in and keeps unwanted visitors or things which are not wanted out. So for instance, that pesky neighborhood pothong that roams along the street, you don't want it getting into your yard. So what do? You have your fence and your gate is closed. Therefore, this pothong cannot or does not have the ability to get in because the fence is blocking him or her. I have nothing I don't have any issues with dogs. I must put that disclaimer in. I just use that as a general comment. So similarly, when we look at the cell membrane, not only does it act as a barrier preventing unwanted things, in particular things like microorganisms and viruses, which could cause harm to the cell, keeping them out, but it also acts as a receptor holder. What do I mean by receptor holder? If we look here, we see a glycoprotein attached to this integral protein here. And what you could have happening, you could have binding at this receptor site, and this could then lead to the opening of channels. So notwithstanding the fact, one of the major properties of the cell membrane is to act as a barrier, it also serves as a recognition device, or holding recognition devices or receptors, which enable specific molecules to enter the cell. So very much like a bouncer at a fete. So you know you have your VIP ticket, right? So if you're a VIP, you approach the bouncer, you show him the ticket, he or her, right, opens the door and lets you in, the special door. And in a very similar way, when you do look at the cell, there are very special channels that allow specific molecules entry into the cell itself. Okay, so after the cell membrane, let's have a look. So here we are seeing in terms of cilia and microvilli, we did mention these adaptation found when you look at microvilli, it's an adaptation that is found, let's say, in cells within the gallbladder, specific cells. And what that does there, it helps to absorb fluid and thereby, by extension, concentrating the bile that is found in the gallbladder. And again, you do have villi or cilia, which do occur, cilia within the trachea, and these cells, particularly the pseudostratified cells associated with the trachea, very important in terms of getting things out. Let's say small particles that go down into your lungs. These trachea, the finger-like projections, in a wave-like action, they remove disturbances or things which could cause disturbances to the lung. They move them out such that it now then comes out of the either through the nasal cavity or through the oral cavity. So very importantly, cilia adaptations to those surface epithelial cells in terms of removal of waste from the trachea, and then the microvilli within the gallbladder, very important in terms of concentrating the gallbladder by increasing the surface area for absorption within the gallbladder itself. So the removal of that liquid leads to a concentration of the bile that is present in the gallbladder. Here we see another structure in terms of cell membrane projections. Here we have flagella, right? The flagellum, which is present on sperm cells. This is a modification which gives the sperm the ability to move, in particular from its site within the vaginal orifice up until the neck of the uterus within the fallopian tube. Sorry, so where fertilization usually occurs. So in terms of that movement, very importantly, the sperm is adapted for that because it has a structure of flagella, which causes the propulsion of the sperm by its movement in a whip-like or wave-like motion. Okay, now here we see the cell itself. Let's see if we could have a look at some of those organelles. The cell nucleus, of course, this is the brain. This is where houses the DNA. DNA has all the instructions for the entire body in terms of its creation. Usually we have 23 pairs, except of course the sex chromosomes, right? They don't have 23 pairs, but they have 23. So the 23 pairs, which are usually found in the somatic cells, very important in terms of they contain all of the information necessary for replication. So that is very important as it relates to the nucleus. Within the nucleus, what else do we see? Well, we see the nucleolus. That is very important for ribosome manufacture. The ribosomes themselves important for protein manufacturing, rough endoplasmic reticulum, so-called, because they're nothing more than smooth endoplasmic reticulum, which has or is studded with these ribosomes. The smooth endoplasmic reticulum, it is important for synthesis of lipids. So that's one of the major functions of it. Golgi apparatus, very important structure associated with packaging of proteins. So the packaging process is facilitated by the Golgi. Cytosol, liquid portion of the cell, very important because the majority of the cell's reactions associated with metabolism need a liquid environment for them to occur. So therefore, the cytosol facilitates these reactions or metabolic reactions that occur within the cell. Then we come to the mitochondria. Mitochondria, as the name implies, might, right, associated with energy generation, energy in the form of ATP, that universal three-letter currency that can be used throughout the cell in order to bring about any type of changes which require energy for its initiation and continuance. You have lysosomes. These are structures which contain lytic enzymes or different types of enzyme which are associated with the breakdown of, let's say, worn out material. That is very important. Or in a unique case, it could initiate a process known as apoptosis, in which the lysosomes spontaneously open, release their contents, leading to the destruction of the cell. It's programmed cell death. And this comes about usually in an instance when the cell detects infestation, microbial or otherwise within the cell, and it recognizes, in terms of the immune system's intervention, recognizes that it cannot save the cell. So instead, what it does, it initiates this pathway that leads to the destruction of the cell, and by extension, the microorganisms or insults to the cell which are causing these disturbances. So therefore, it causes destruction in a localized region, thus preventing the spread of the microbe, let's say, to other parts of the tissue, and by extension to other cells as well. Cytoskeleton and centriole. Centriole important, as we will see later on, as it relates to division, both in meiosis and mitosis. Right, mitosis, which leads to the production of identical daughter cells, and meiosis critical for the production of haploid cells, which are needed in terms of the male sperm and in the female, by extension, the ovum, and these are critical for the reproductive process. Peroxisomes, they do contain peroxide, and they are important for the oxidative digestive processes that occur within the cell. And I think I have addressed, the vesicles, these could be used for storage, of course, within the cell. So just to highlight those structures which were mentioned, the Golgi apparatus, cytosol, liquid portion, cell membrane, it's a structure which it allows things to come in and keeps all those unwanted ones, mitochondria, energy, nucleus, brain, the nucleolar site of production of ribosomes, ribosome, protein synthesis, rough ER, nothing more than endoplasmic reticulum studied by ribosomes, peroxisome, important for oxidative digestion, and the smooth ER, important for lipid synthesis. Okay, so these are the things which were previously mentioned in terms of what is found within the cell itself, in terms of DNA, the endoplasmic reticulum, Golgi apparatus, mitochondria, cytoskeleton. All of these things are very important. Okay, so let's look at reproduction. One of the major things associated with the cell itself is the whole notion of cell reproduction. And this is not only something that is true for cells, but it's true straight across the entire biological world. In fact, one of the major reasons that organisms are here, and by extension, when I say organism, I refer to complex and simple ones, is to reproduce. Because if you stop reproduction, then you will disappear as a species, you will become extinct. So therefore, it's very critical, this process of reproduction. So you do have apoptosis, as I mentioned, natural or programmed cell death, which involves a cascade of reactions within the cell itself, leading to the opening of lysosomes and the release of enzyme, which then leads to the cell death. Necrosis is cell death due to injury or tissue, and again, this could be initiated by similar mechanisms in terms of lytic release of enzymes, which could cause cell destruction. All right, so we spoke about the destruction of the cell, we spoke about the components of the cell, and we spoke about maintenance from the perspective of destruction of the cell. Now let's look to the cell cycle. How does reproduction occur within the cell? Well, in this case, we're looking at mitosis. Mitosis, as previously mentioned, produces identical daughter cells. So for instance, when we think about our skin, when we do take a bath on afternoons, and we do see a little bit of gunk or dirtiness that comes off, not only is it dirt, but also dead cells which come off. So how are those dead cells replaced? Well, they are replaced by this mitotic process. And let's see if we could go through this mitotic process and explain what is happening. Principally, mitosis occurs or consists of major parts: prophase, metaphase, anaphase, and telophase. There's also the part which takes the longest, as you see here, 22 hours to many years. This is interphase. And during interphase, you have three processes that occur: G1, S, and G2. G1, this is when organ duplication occurs. S is DNA duplication. And G2, you have growth of the cell, because the cell has to get larger because it has to go through a process in terms of division. Then you have prophase, metaphase, anaphase, and telophase. And what this is leading up to, particularly metaphase, you have alignment of the chromosomes along the center of the cell, or the equatorial region. Anaphase, and you have the divided chromosomes moving to the separate sides of the cell itself. Then you have telophase, in which you have cytokinesis occurring. Cytokinesis ideally is referring to the separation of the one cell now into two as it relates to the cell membrane. So now you have the formation of two separate distinct cells, and that is cytokinesis, and that usually marks the end of the mitotic process. So DNA replication. This DNA replication has occurred during interphase S phase of interphase. And what does it involve? Well, it simply involves, one of the unique things with DNA is that it's a double helix, and it has the ability then to unwind, and by complementary base pairing, you could then form a copy of the strand that has unwound. So in terms of the mechanism for replication, it occurs primarily by unwinding of the DNA double helix, and then through the intervention of different proteins coming in, you do have base pairing which does occur, and this base pairing leads to the formation of new strands, right, off of the template strand. And of course, this occurs under the action of different proteins, DNA polymerase, DNA synthase, DNA helicase, which needs to unwind uh, the, the, the tightness or and also by extension the kinks which does occur. The DNA helicase is very important in releasing that tension which exists in the strand. Let's get back to mitosis. As we mentioned, interphase, longest phase consisting of G1, S, G2. During G1, organelle distribution, duplication, during S, DNA replication, and during the G2, you have growth occurring. Prophase, metaphase, anaphase, and telophase. Prophase, here we are seeing them, the spindle fibers attaching to the centromeres. Chromatids are moving towards the equatorial region of the cell. As it's seen here, they line up for metaphase. Anaphase, you have a furrow forming, and the chromatids are now moving towards the end, and then towards either pole. And during telophase, you have now cytokinesis being completed, and two separate and distinct daughter cells. Note well for mitosis, these are identical copies of each other. All right, so stem cells, very important, stem cells. These are the cells from which all other cells are derived. And you have three different classes, totipotent, pluripotent, and multipotent. The first one, the totipotent stem cells, these could give rise to an entire organism. The pluripotent ones, they could only produce any type of tissue, but it can't produce the entire organism. Whereas in multipurpose, they can only produce a limited range of cells. And all of these stem cells are important when you do look towards the whole notion of fusion of male and female gametes to give a zygote, and then ultimately for the specialization of these cells throughout the body, giving an entire human. So here we have the replication process, giving the formation of a zygote as shown here. And then you have, by repeated cell division, you have the formation of a set of cells, and some of them are totipotent stem cells, as we mentioned. These are the unique ones which have the ability to become any cell that they desire. From here, they move to pluripotent. Pluripotent are very specialized for the formation of certain structures, and then you with continued differentiation, you have the formation of different muscle types, different cell types from these multipotent stem cells. So this is very important, the whole notion of cell differentiation from the totipotent set down into the multipotent stem cells. All right, so that explains how you could go from just the fusion of two cells, zygote and ovum, and how you could get the formation of an entire human because of the fact the stem cells are present, and then they differentiate repeatedly in giving you different structures within the human body. And when we say structures, we're referring to tissues, muscles, bone, organs themselves. So how is this facilitated? Well, as we mentioned, everything comes off of the DNA. The message has to be moved from the DNA, which holds the transcript or all of the instructions for the entire human. And to do that, RNA is the carrier. So one of the things you have to appreciate, as is written in the DNA central dogma, is DNA is transcribed into RNA, right? So the RNA goes through the nuclear pore, into the nucleus, copies the message, it then leaves via the nuclear pore, gets back into the cytoplasm, and then this message is translated into proteins. So the whole concept of DNA transcribed into RNA and then translated into protein, this is referred to as the central dogma. Do note that you could have DNA replication, of course, and so you could have DNA selfing on itself in terms of making more DNA. And then this third portion does complete the triad associated with the DNA central dogma. So RNA, let's talk about the types. We have messenger, transfer, ribosomal, and small nuclear RNA. And in terms of their functions, the messenger, they act as a template for protein synthesis. They transfer amino acids to ribosome for protein synthesis. right? The ribosomal, they fold the proteins together, and the small nuclei, they form part of the enzymes that modify the RNA. So the messenger and the transfer, these are the two important ones. The messenger is the one that actually copies the message, and the transfer, these bring specific amino acids to the ribosome for protein synthesis. So this is just showing the portion, how it happens. So here you have the double helix of the DNA, right? And now you have the RNA coming in, and by complementary base pairing, it copies the message of the DNA of the DNA template. Now, of course, in terms of the message, this could be a particular gene, and the gene leading to the production of specific types of protein. So therefore, for the production of the protein to occur, messenger RNA has to be transcribed off of the DNA. Then it goes into the cytoplasm, where it then hooks up with the with the ribosome, and then from the ribosome itself, you now have the formation of the protein, right, via the tRNA bringing in the specific amino acids. Of course, we did look at the fact that proteins themselves, they are composed of amino acids in joined together. Okay, so to appreciate, now, one of the important things we have to recognize for all of these things to occur seamlessly, it really is one of the fantastic goals or things which happens at the level of the human in terms of from the gene being transcribed and then translated into protein. Sometimes, however, things do go wrong, and cystic fibrosis is one of those examples. Let's see if we can have a look and see what happens in this case. Well, for cystic fibrosis, you do have gene deletion of the CFTR. And you might be saying, what is the CFTR? Well, the CFTR is a cystic fibrosis transmembrane conductance regulator. So the CFTR is a specific protein important for the formation of normal mucus within the lungs itself. So therefore, with cystic fibrosis, when you do have this mutation occurring because of this of the on this protein, what it causes is a thicker mucus to develop. And this thicker mucus does lead to congestion within those who are so afflicted with the disease. And this thicker mucus leads then to issues relating to normal breathing patterns. So let's see how this occurs. Well, um, it occurs because here you have the CFTR gene, and on the CFTR gene, the normal case, you have this being present in terms of these the CGT on the sense template, DNA sense template, right? The sense strand, but if you look on the template itself, which is the lower one, you have GAA reading from left to right, right? So reading from left to right, you have a GAA, and this is deleted. So when you look at the cystic fibrosis gene in terms of the persons who do have the mutation, GAA is missing. And this is amino acid number 508, which codes for phenylalanine. This is missing when you do look at the gene present in cystic fibrosis sufferers. And because you have that deletion, so you don't have phenylalanine being present, it causes this issue. So in terms of what does this lead to? So if we were to look here, the gene in terms of the normal occurrence of the gene itself, right, you would have it being transcribed and then translated within the cytoplasm. But with cystic fibrosis, in terms of the proteasome or the set of protein or the set of amino acids which comprise a specific protein, right, what you would have happening here is you would have one of them missing in terms of that. So what does it cause? Well, it does interfere with the movement of substances across the membrane itself. And we get into a little more detail as how it specifically affects cystic fibrosis sufferers. But for now, let's talk about membrane transport as it relates to these three actions: diffusion, active transport, and selective permeability. Now, diffusion is simply a movement of substances from its area of high concentration to low until you have some type of equilibrium being established on either side of the membrane. Active active transport is movement up a concentration gradient that requires energy. This energy is usually in the form of ATP. Selective permeability is you have lipid-soluble substances. They can course freely across the membrane because the membrane is primarily composed of lipids. It's a lipid bilayer, but others require escort from membrane proteins. So therefore, selective permeability, only certain ones can pass through. Let's talk about simple diffusion. And this occurs all the time. This is what allows, if you are in a room and somebody comes in wearing perfume, for women, cologne for men, this is why you smell it, because the concentration of these molecules are high in terms of on the person. They are odorants, so they do have the ability to spread off of the clothing or the skin of the person. Now, these odorants, which were previously in low concentration in the room or absent altogether. So they move from the area of high concentration on the skin or from the clothes of the individual to the area of low concentration within the room itself, the air. And therefore, it is detected in your nasal cavity by those receptors and perceived as a certain type of smell in your brain, in the olfactory region of the brain. So very importantly, as shown in this case, cookies, yum. They are smelt, and the young girl smell these cookies. She gets an odor of it, right? So that's how it is smelled, because you have simple diffusion of these chemical odorants from the cookies to the nasal, to the nose of the young girl in this case, right? So, and then the olfactory, it is processed in the olfactory portion of the brain. So that is very important. So facilitated diffusion, this refers to, so in the excess and the movement down a concentration gradient, but it requires a certain channel for it to pass through. So in this case, a sodium channel is passes, allowing selectively for the only sodium to pass through. In this case, down its concentration gradient. Active transport, a very similar mechanism, but in this case, it doesn't go down its concentration gradient, it's moving against its concentration gradient. And it does so using ATP, so using extra energy to move it against its concentration gradient. So that's very important as it relates to active transport. All right, so osmosis is a special case involving movement of water from its area of high concentration to its area of low concentration across a semi or selectively permeable membrane. And this measure is done by osmolarity. And what osmolarity refers to, the number of particles of solute per liter of solvent, right? And this is how it is measured. All right, so let's see what happens in the blood as it relates to osmosis. Now, normally, if a blood cell, here the red blood cell, is in an isotonic medium, so that means then the concentration of water within, or the concentration of solute, both inside and outside of the cell, is virtually the same. So therefore, what you'd have happening, an equal amount moves in and out, so the cell itself keeps its form. What happens when it's put in a place in a hypertonic? Hypertonic means that the solute concentration is higher on the outside. So therefore it means the water concentration is relatively low as compared to the inside of the cell. So therefore water moves down its concentration gradient, high within the cell to outside, low on the external environment. Water moves out of the cell, causing the cell to shrink. And by extension, you have an opposite happening in a hypotonic environment. Hypotonic means that you have relatively little solute, so therefore the water concentration is particularly, in this case, it is very high compared to the inside. So inside is lower, concentration of water is higher on the outside, so water moves down its concentration gradient up from outside the cell to the internal environment of the cell, causing the cell to swell as shown here. So it looks like a balloon filled with water. So how does this relate to cystic fibrosis? Well, chlorine exits via the CFTR channel, usually creating an osmotic gradient. So here, often you see chlorine is leaving the cell, and this is, of course, this is the mucus within the normal airway. So here, this is showing normal mucus. notice a thin amount of mucus, and this is kept in place because of the fact of the movement of chloride ions, because when the chloride ion moves, one of the things that happens, water moves as well. And this, when the water moves, it keeps the mucus relatively thin so that it could move out of the passage airway. Now, with cystic fibrosis, the channel does not exist, so therefore chlorine cannot move out, and by extension, the water to follow chlorine. So what happens then, you do not have a dilution of the mucus, and it is relatively thick, and it builds up, as we see here, constricting the airway, and therefore persons, as in this case named Julia, she would have difficulty in terms of breathing because of the thickness of the mucus itself. All right, so that is what specific case of cystic fibrosis. Let's speak about exocytosis, which is the ability of the cell, or refers to the transfer mechanism associated with cells in which vesicles fuse with the membrane and deposit their contents to the external environment. Endocytosis is just a reverse of exo. So what you have, invagination of the membrane, which then allows for things to be taken in. So the membrane then seals off and forms a vesicle, which then could either one, be then fused with a lysosome and leads to destruction, or two, it could just remain within the vesicle, just as a storage unit, as it were. Phagocytosis, let's talk about that. This refers to cell eating, and specific cells have the ability by an endocytotic mechanism to take in this case, as is shown, a bacterium. Once the bacterium is enclosed in the vesicle within the interior of the phago, in this case, looks like a white cell, a T cell. Once it is enclosed there, you could have then fusion of the lysosome with this vesicle, and then this fusion of the causes the lytic enzymes to then lead to destruction of the bacteria. Okay, so those were the mechanisms involved with transportation of substances within the cell, and also to remove substances out of the cell. Let's talk about the tissue types themselves. So there are four basic tissue types: connective, muscle, nervous, and epithelial, and they are distinguished by the cells and the matrix extracellular that let's see if we could differentiate them. So you have epithelial cells. They're usually flat, have multiple layers, and they line the skin, esophagus, and vagina. So they're shown here in terms of epithelial cell, and their main function is one of a protection. Then you have here, that also shows some more stratified cuboidal epithelium, and this is transitional epithelium, and these usually line the urinary bladder wall. They are multiple cells, and they allow distension. All right, so very much like a bag, when things are put in them, they have the ability to extend. Hence the reason that they are found in the bladder, because the bladder does need distension because of the fact that it could hold variable volumes of urine depending on the situation. Cell junctions. This is an adaptation of the cell in terms of providing a cutoff, or an ability to actually join to other cells as well, not only as a cutoff mechanism. And these consist of tight junctions, desmosomes, and gap junctions. The desmosomes, they anchor the cells together like rivets. The tight junctions, they seal the cells together, preventing movement of fluids between them. So that's how they cut off things from passing. Whereas the gap, they function in communicating by allowing small molecules to pass through them. All right, then we have glands. Very importantly, we have exocrine and endocrine glands. Exocrine, they secrete their substances or their secretions to outside of themselves in terms of external environment, whereas endocrine, they are intimately associated with blood vessels, and they secrete their substances into the blood vessel, which then carries it throughout the body itself. And the last thing we'd want to address is connective tissue, right? So you have loose connective tissue. For example, they're web-like and they also could store fat. This is like adipose tissue. And you have dense connective tissues. And these are important for scars, ligaments, and fascia. You have specialized connective tissue like cartilage. You have the hyaline cartilage. You have fibrocartilage and elastic cartilage, very important. And you also have bone and blood, which blood, oftentimes you don't think about it, but blood is categorized as a special connective tissue as well. Okay, so that concludes this lecture. And what we looked at, we looked at the function or the structure of the cell. We look at the functions of the different parts of the cell, from the nucleus to the different organelles within the cell, the importance of what they do. We moved on from there, and then we looked at the process of how things move within the cell by diffusion, movement along a concentration gradient, or facilitated, in which you could have movement against a concentration gradient. But what is critical is energy to do that movement across the concentration gradient. Then we spoke about cystic fibrosis. Cystic fibrosis occurring because of that mutation on or deletion of amino acid number 508, right, of phenylalanine. And of course, what that leads to is the formation of a thicker mucus in your air passages. And this thicker mucus, of course, leads to the sufferer. It causes them issues as it relates to breathing. All right, so cystic fibrosis shows the importance of actually copying that template, DNA template correctly. So this, of course, does usually happen. You do have mechanism in place to actually double check it. But in certain instances where the mutation does carry over, it does shows then one, just a simple mutation, a deletion mutation, how it could have dire consequences in the sufferers. Then we mentioned the different types of tissues and their locations. Again, very important, showing that link between structure and function. All right, so that is it for the lecture for today. Hope you did enjoy it, and I'll see you again later on. Okay, have a good one. Good afternoon.